Thanks for the introduction. I will present some results of mercury observations and modeling during the NOMADSS campaign, and how do they improve our understanding of mercury emission fluxes from the land and the ocean. The mercury measurements were made by Dan Jaffe's group at Univ Washington during NOMADSS, and Jaegle's group and UW and Selin's group at MIT works on interpreting these mercury measurements using chemical transfer models. We also acknowledge the authors who made the VOC measurements using TOGA and PTRMS during the NOMADSS. So this cartoon shows an estimate of current global mercury cycling, and we can see that most of the mercury is emitted into the atmosphere in the form of Hg0, which is gaseous element mercury, which is also called GM. GM has a lifetime of several months against oxidation to reactive mercury, Hg2, which returns to the Earth's surface by deposition relatively quickly. Therefore, it is very important to understand the atmosphere surface change of GEM because it drives the mercury cycle. We can also see from the figure that the GEM emissions from the land surfaces are higher than over the ocean surfaces. Also, considering that the land surface only accounts for 30% of the Earth, while the ocean surface accounts for the left 70%, that the average GM emission per unit area from the land will start to exceed the ocean. Therefore, that we would expect that the GM concentrations over the land surfaces should generate higher than over the ocean surfaces. Previous model simulations are consistent with such understanding. This figure shows the modeled surface GM concentrations over North America and Western North Atlantic from several global and regional chemical transport models. These models have very different resolutions and different chemical mechanisms, but they all predict higher GM concentrations over the land surfaces than over the ocean surface, which is consistent with our understanding that land emissions are higher than the ocean emissions. However, recent observations in North America have challenged such understanding. Atmospheric mercury measurement network called AMNITE was established in 2009, which involves dozens of surface sites measuring GM concentrations. David Gay et al. Summarized GM data from 2009 to 2011 in AMNITE, and they combined these surface sites into several groups based on their locations. An interesting finding in their study is that the continental remote group has about 0.1 nanogram per cubic meters lower GM concentration compared to the coastal remote group, which is in contrast to our understanding that land emissions are higher than ocean emissions. This 0.1 nanogram per cubic meters of GM is kind of substantial because the lifetime of GM is several months in average, and the concentration level of GM is about 0.1 nanogram per cubic meters. However, no definitive conclusion can be drawn from this plot because of limited measurement size, and more important, the large intercomparison errors of GM observations by different labs. This plot shows results from two intercomparison experiments of GM observations. We can see that GM concentrations measured by different labs can differ by 0.1 to 0.3 nanogram per cubic meters. This large intercomparison error comes from different labs follow control operational standard and calibration actual. In our recent work, which is called top-down constraints on atmospheric mercury emissions and implications for global biogeochemical cycling, which is under review in ACPD, we collected GM concentrations from 27 surface sites around the globe and quantify mercury emissions and several parameters in geoscale model using an inversion approach. This inversion approach can take into account both errors in emissions, parameters, and observations, including the previous intercomparison errors. The the results related to this NOMADSS work is that the inversion increases the GM emissions from the northern hemispheric ocean, but decreases the GM emissions from the land. We will see the effects of these changes when comparing with the NOMADSS environment data. The NOMADSS campaign provides new constraints on the atmosphere surface change of GM because only one set of instruments by Universal Washington is used, and therefore intercommercial errors are avoided. And second, it covers large spatial areas. We can see we measure GM concentrations both over land and over the ocean. And third, many other tracers are measured simultaneously, which may provide additional constraints. Because we study the atmosphere surface change of GM, we selected background GM data in the pre-boundary layer using several criteria, including potential temperature, altitude, water weight mixing ratios. We've also removed plume events by concentrations of CO, SO2, NOx, etc. We further divide GM concentrations into two categories: over land and over water measurements. We use the GeoScam model to interpret these NOMADSS measurements. GeoScam is a global chemical transponder model with finite resolution over North America. We use the GeoScam for chemistry simulations to model VOC tracer measurements. And for offline mercury simulations, we perform two model cases. As we said, one uses standard configuration, we call it the standard case, and the other applies our top-down constraints from global GM observations, we call it the inversion case. Now we are seeing the comparison of GM emissions between the standard and the inversion simulations during the NOMADSS campaign 2013 June to July over eastern US and western North Atlantic. The figures show spatial distributions of mercury fluxes, and the table shows the total emissions over the domain. We can see that in the standard case, both the ocean and the soil and vegetation system are net sources of GM. They contribute to similar emissions. And in the inversion case, the ocean becomes a larger source. The total emission doubles. But the soil and the vegetation system become a net sink of GM. We do not change emissions of we do not change unspent emissions, geogenic emissions, and biomass burning. Now we are seeing the observed GM concentrations during NOMADSS and the model results from two model cases. As I said, we separate the measurements into two categories: over land and over water. The boxes, the red color shows observation, blue color shows results from standard simulation, and the green shows inversion simulation. The boxes shows 10 percentile, 90 percentile, and the median. We can see that the observed overwater GM is 0.12 nanogram per cubic meter higher than overland GM, which is significant. This result is consistent with with the measurements from Ammonite network. However, the standard simulation shows higher overland GM than overwater GM, which is in opposite way. After the inversion simulation shows comparable GM between these two groups. Therefore, that we can see that the inversion simulation better reproduce the land-ocean difference of GM concentrations from the NOMADSS campaign. However, we can see there is a 0.2 nanogram per cubic meter difference between the concentration level simulated by the inversion case compared to the NOMADSS observation. We hypothesize that the interannual variability of GM concentration can contribute to this difference. The left figure shows the locations of 4 ammonite sites, and the right figure shows their observations in 2013 summer, 2009 to 2011 summers, and the model results from inversion case. We can see the inversion simulation has no bias compared to GM observations during 2009 to 2011 summers, but we found that the observed GM concentrations during 2013 summer are about 0.12 nanogram higher than the 2009 to 2011 summers. We are not sure about the reason for this interannual variability yet. They may become meteorological or chemical factors. As I said, because during the NOMADSS campaign, many other tracers are measured, so which may provide additional constraints on mercury emissions. This slide shows correlations of GM with biogenic and ocean VOC tracers. DMS is the ocean tracer, and aspirin and monoterpenes biogenic tracers. From the observations that we can find, there is a significant positive correlation between GM and DMS, but negative correlations between GM and two biogenic tracers. This may suggest that DMS is this may suggest that the ocean is a larger source than what we thought, while vegetation could be a very large sink of GM. While in the standard simulations, while the standard simulation shows opposite correlation relationships compared to observations, basically that's the model cannot reproduce such correlation relationships. The invariant case shows weakly correlations between GM and VOC tracers. So those are preliminary results, and we are working on further adjusting the emissions and model parameters to reproduce those measured correlations. So, in summary, that's the recent observations in North America have challenged our previous understanding of the land emissions and ocean emissions of mercury. Applying our constraints from global mercury observations can better reproduce the land-ocean differences of GM concentration during NOMADSS. The land surface of eastern US is a net sink of GM, while the western North Atlantic Ocean is a large source of GM. These results are supported by correlations between GM and biogenic, oceanic, and ocean VOC tracers. Future work includes examining several other processes, for example, emissions of GM from coastal regions, which is a missing process in geoscience model, and uncertainties in unsustainable emissions. So I will stop here, and thanks for your attention, and I'm happy to hear any questions.
Time for a few questions. Daniel?
So we know for some time that the North Atlantic Ocean contains very elevated mercury levels, and that's in some versions of geoscan. And I was wondering if your standard model included that, and the North Atlantic level increased even more than we thought?
I think what you mentioned, that's in the 1930s to 1980s, because of the input of from the rivers into the ocean, that's the intermediate ocean contains higher higher mercury concentrations. But I think currently not in the standard geoscience model, that's the concentration in North Atlantic is really low.
Yes, Jenny.
When you showed your inversion results, you showed change to the gross soil term.
Yeah.
Can you change the soil globally in the same direction everywhere, or are there spatial differences due to different vegetation types?
In the previous invariant work by global GM concentrations, that we use a single factor around the globe, not specific, because we don't have reason to do that.
So I have a question. The anti-correlation between GEM and biogenics is interesting. Now, in NOMADSS, there were a lot of racetracks at different levels in the boundary layer and above. Is that useful data to constrain the flux?
I think one difficulty for mercury is that we are quite uncertain about the oxidation of GM in the free troposphere. So because of this large uncertainty, it is very difficult to detect whether this is due to emissions or chemical reactions. Yeah.
My first thought about this is that it's deposition of elemental mercury. Is it underestimated in the model?
Yeah, it could be. Yeah, This we haven't tested to use different to use a different gravitational velocity of element mercury, but yeah, we are planning to do that. Yeah.
Okay, thanks. We have to move on. Thanks again.